right, so my name's Jose Zuniga. You guys already know that. This is Jess Tompkins, a very good friend of mine. What university did you go to?
I went to Monash University in Melbourne, Australia.
And what was the degree you were pursuing there?
I'm actually still in it. I am pursuing a Bachelor of Science majoring in immunology. I've got one semester left.
So she's qualified. All right, so we are going to go over the 14 different topics that we have for the project, but we'll try and do it as concise as possible. So, what is microscopy? Explain it. Microscopy is the technical field of using microscopes to view objects and areas that cannot be seen with the naked eye, and viruses are typically viewed with electron microscopy because they're like so small. Mm-hmm. And how do you think this ties in with virology?
Well, obviously the study of virology, we're looking at how viruses impact, particularly human cells, and you can't see that with the naked eye or on a macroscopic level, so that's why we use the electron microscope to go into that deeper level. And so you can see how viruses are interacting, with surface proteins, and also like the different shapes and stuff. Yeah.
Mm-hmm. Yeah, because there's some that are like, here, let me think off the top of my head. There's like icosahedral, pleomorphic, there's helical, and then obviously if it's naked or if it's enveloped.
Mm-hmm.
All right, so the second question, what are organelles? All right, so organelles are subcellular structures that are used to carry out important jobs like an organ would in your body. Some examples, in like a cell, there's the nucleus, there's the Golgi apparatus, the ER, vesicles, and the mitochondria, ribosomes. And how do you think that ties in with like viruses? Like what specific organelles would they need?
Yeah, well, because viruses are, non-cellular obligate agents, they don't have their own machinery, they don't have their own organelles to replicate and, perform their function of life. So that's why they infect human cellular organisms, and then they invade and use their organelles to perform their functions.
Yeah, because they can't make proteins, so they use the ribosomes.
Exactly.
They can't make ATP, so they use the mitochondria. And
obviously the nucleus, they need that to
for the genetic material to steal the identity.
Literally everything, they basically just take over and use everything possible.
Yeah, honestly, it's kind of awful, I'm not gonna lie.
It's kind of cool.
Yeah. Alright, cool. So the next one is going to be, what are enzymes used for in a cell? So within like a healthy cell, enzymes are proteins that help speed up the metabolism or chemical reactions within the cell. And they can help build some substances and they can break them down. But I typically know them for things that break it down. So how do you think this could tie in with viral entry?
Well, yeah, you see it a lot specifically with bacteriophages, which are viruses that infect bacteria, and they actually use lysozyme, which is found in lysosomes. So obviously the recycling and degrading sort of organelle, they can capitalize on that and use that to actually degrade the membrane of the bacteria to help them infect and inject their own nucleic and genetic material into it.
And then, like for example, one that I was thinking of. How can we use the enzymes to our advantage? Like Ebola, it uses a glycoprotein on its surface so that it can attach to a host cell, and then once it's inside, it's macropinocytosis, so it engulfs it, and then it gets put in a lysosome. And then there's a protein in the wall called MPC1, and it needs to fuse with that protein. But if it doesn't, the digestive enzymes inside that lysosome eats up the Ebola virus, so then it just gets metabolized. So if they're actually a drug that blocks NPC1, so then it keeps it in there. Yeah, so we can use it.
And then it just gets by the natural body. Okay, so next we're going to talk about cellular structures in animals and plants. And obviously all cells are encapsulated by a cell membrane, and in the case of plants, a cell wall in addition. But Jose, I was wondering if you could actually tell me whether these help or hinder a cell when it is attached to a virus.
So for plants, the short answer is no. viroids, small pathogenic RNAs. They have no protein code, cause virus-like diseases in plants. This is what viroids are. And they're these small channels that connect the plant cells to one another called plasmodesmata. And these viroids have this gene called a movement protein. And basically, it helps them form tubules within the plasmodesmata so they can move from cell to cell in between the channels. So, like, if anything, it probably helps it out. Yeah. So, what is DNA and RNA? These are pretty basic, like ideas. DNA and RNA, they're genetic materials that a cell can read in order to make proteins that have specific functions to form organelles, and then as it gets bigger, cells, organs, so on and so forth. How can we identify cells through their DNA or RNA?
Yeah, well, lots of scientists use a procedure called PCR, or polymerase chain reaction, because the genetic material available naturally is so small, so we need to find a way to amplify that. And so that's done through three main steps, where first you take your DNA and you split it through denaturation, and then you have something called annealing, which is where you have primers that are attached to the ends of that separated DNA, and they extend using the nucleotides to create, so that essentially at the end of one round, you've got two new sets of that one amplified DNA, and if you just continue to go that over, then you're going to have a large amount of DNA for your analysis that you can see based on size and charge if you use gel electrophoresis.
Yeah, damn, that's really spot on. That's really good. It's at the halfway point.
Yes. So, what ties do transcription and translation have with an RNA or a DNA virus? Well, because viruses need a host cell and to use its machinery to, undergo protein synthesis and export its proteins for whatever function of life it requires, as well as replication, it has to use that host cell. And this actually also depends on what type of virus it is. So whether it's positive or negative sense, single-stranded, double-stranded, RNA, DNA, all of these things impact what processes it needs to undergo and what polymerases it needs to use to perform whatever it needs.
Yeah. So for the sense of the positive sense, those ones can code for protein right away, and then the negative sense, that's a template for positive. And then if you have something that's double-stranded in a eukaryotic cell, I think it would be all right. But if it is a double-stranded that's fragmented, since it's broken up in pieces, it needs to go to a singular piece, so it goes from that fragmented to a whole piece of RNA, and then it goes back to whole DNA. Yeah, and with RNA virus, every single one needs a polymerase because eukaryotic cells are monocystronic, so they code for the one gene. When viruses, they have multiple different genes, usually 3 to 5 is what they have. So the next one that we have is can protein sorting affect a virus if it is segmented? And protein sorting is the biological mechanism by which proteins are transported to their appropriate destination within or outside of the cell. And a short answer for this is that there's pro and cons, and we can discuss that.
Yeah, so I would say an advantage of having a segmented virus is that all of its genetic material can be encoded in a single particle, is housed in a single particle, and this protects it from breakages, especially due to shearing, and so obviously it's more protected and it increases the total potential output of coding capacity.
Okay, cool. And so even though that's really good, I think the disadvantage of it is that all of the individual genome segments, they have to be packaged in that one particle. And if it's not, there's going to be a defect and it's going to be a loss of genetic information. And if there's a loss of genetic information when it comes to the protein sorting, it's not going to have all the instructions it needs to send something to a specific place to make the virion, or if it doesn't have the information for a specific protein, it's not going to be made or moved. And that's where I think mutation would come into play there, where it would be a different virion if it was able to be completely completed and sent out.
Yeah.
This has been so much fun.
It's going well.
I know it is.
Okay, so we know that vesicles are organelles that help transport materials within and inside and outside of cells, but I was wondering, how can we compare this vesicle transport to the use of viral vectors and vaccinations?
Okay, cool. So vesicles can help transport material to organisms that need it for survival, and they can recycle the waste like you just mentioned. And so I think for viral vectors on the counter side, we can use them for gene therapy. So you take a virus and you weaken it so that it can't harm the patient, so it's considered attenuated. And you insert a correct gene that will fix the disease. And then this virus is inserted in the patient, via vaccine. And a really good candidate for this is cystic fibrosis because it only has one gene that needs to be fixed. And we already have prior knowledge as to where this gene is, so it's super easy to target. But yeah, it's pretty cool how a vesicle moves stuff around, and then you can take a virus, put a gene in it, and then it will move inside the body to fix for gene therapy. And that's pretty cool.
So can viruses actually make their own ATP?
So viruses are not capable of making their own ATP. They need the host cell's mitochondria to make ATP to further, go through the replication process. And a host cell undergoes ATP hydrolysis. So this is the release of energy present in high energy terminal phosphate bonds, which is utilized to carry out the rest of it. So the actual breakdown. So the phosphate group, it doesn't want to stay positive, so it takes electrons from the H2O to become more negative while breaking off of the ATP molecule, and that is the energy release that it can use for work. So how do you think, or is metabolism a danger to a virus or a vaccine?
Yeah, well, I actually think this is really interesting because having a high metabolism is just a natural source of immunity for us. Because when a virus does enter a cell, it is engulfed, via endocytosis, macropinocytosis, and it is encapsulated in a vesicle. But as we spoke about before, there are enzymes in these vesicles, in lysosomes and stuff, containing, digestive enzymes, and so viruses may not ever reach their destination if they are encapsulated by these vesicles because they're just degraded naturally from a high metabolism, which is really helpful.
But on the counter side, if a virus is used as a vector for gene therapy, you want the virus to make it through so that it can hit its target. But then if the body just metabolizes it and it sees, okay, this is an invader, we need to take it down, it's doing its job so well that it doesn't get the actual treatment, which is but yeah. And then on the top of the thing with the lysosome, so typically if it does enter like that, the virus would fuse with that membrane of the lysosome so that it can't get the genetic material out. But in the case that it doesn't, that's when it would be digested by all the enzymes. The cell signaling. Okay, do viruses benefit from the cell signaling pathway?
Well, yeah, I would say yes. So as we know, there are many methods of intercellular communication. So you've got the endocrine system, paracrine system, all that sort of stuff. But in the case of viruses, they actually take advantage of direct contact. So most viruses have some sort of ligand, like a glycoprotein or spike protein. With COVID-19, the spike protein was what everyone was talking about. But basically, that is what interacts with the cell membrane that they are trying to invade. And so then this interaction is what stimulates the macropinocytosis for actually to be engulfed, and from there, that's where it undergoes all of its life functions and replication. So I would say yes.
Yeah, not for sure. But, so what is this? A form of receptor. A form of receptor on the surface of a cell can be a G-coupled protein receptor. So this one goes in and out of the membrane 7 times, and it can be used to onset reactions within a cell. So something can attach to it, and then the cell's gonna be like, oh, we need to do something.
Okay.
Yeah. All right, cool.
Cool. So how do cancer and virology tie in with one another?
So they are in a very, very close relationship. So oncogenes are growth factor genes that can be mutated by a virus when it integrates itself into the host cell. And this mutation can make the cells grow abnormally and then multiply, and this is what causes cancer. And what actually stops this happening in your body is the tumor suppressor gene, because think about it, you get so many different viruses, illnesses all the time, but what stops this is the tumor suppressor gene. But if the tumor suppressor gene gets mutated, then it can't turn itself on. It's been completely changed, and then that is when
the oncogenes
they can just keep growing and growing, and nothing's going to stop it. Wow.
And are there any viruses that can actually affect muscle contraction within the body?
I'm glad you asked. So I actually just learned this. I looked it up. Smooth muscle contraction can be caused by hyperactive airways that influenza may give to a patient. So this is why the influenza virus can give you a sore throat and a steady cough. And the influenza virus is an RNA virus, so it's constantly mutating every year. So this is why we always have to get a vaccine every single year for it, instead of just getting one vaccine, which what you would get with the DNA virus. And there are so many mutations because of the lack of proofreading that our RNA polymerases have.
Right, so would you say that's the same for COVID-19? That's why we need to keep getting immunized against it?
So all the different ones like Omicron, and then there was like SARS.
Yeah.
So these are all mutations that are happening because of its lack of proofreading.
Right,
So it's
okay.
really hard for the CDC.
Good job.
All right, now this is over. Shout out Brett Clark, shout out Dr. Horn, shout out Valley Burnett, Tyler Nungasser, and shout out Lindsay Bowen.
He was an A, he deserved